Today's webinar is Introduction to RNA-Seq Analysis. This is part 4 of our 4-part RNA-Seq Webinar Series. You can find recordings of part 1, 2, and 3 on our support site. Part 1 mainly covers an introduction to RNA sequencing. Part 2 discusses the kits that you can use, and Part 3 will discuss best practices. Okay, so let's jump into our Introduction to Analysis. Here is our agenda for today. First, we'll discuss the main goals of RNA-Seq analysis. Typically, when we perform RNA-Seq experiments, we're looking to find if genes are differentially expressed and by how much. Another area that we can look into is novel transcript assembly, and we may even want to perform both DE and novel transcript assembly. Then we're going to go over some of our workflows in BaseSpace that will enable us to perform both. After that, we're going to cover our newest available workflow for RNA-Seq analysis in detail. This includes an in-depth look at the RNA-Seq Alignment version 2.0.1 app, its methods and output files. After that, we'll detail the RNA-Seq Differential Expression version 1.0.1 app and learn how the RNA-Seq Alignment output is used in this analysis. Lastly, we'll discuss the RNA Custom Genome Builder in some detail for those of you that are interested in performing RNA-Seq analysis with a custom genome. We'll then conclude today's presentation with an overview of other RNA-Seq alignment tools on BaseSpace that can also be used for RNA-Seq analysis goals. Okay, so let's kick off by defining differential expression analysis and novel transcript assembly. So, differential expression analysis, what is this? You can think of it as a quantitative comparison of the gene or transcript counts between 2 or more samples or groups of samples. It's a way of identifying the genes that are differentially expressed under 2 or more different experimental conditions. So you have options for what you're looking at when you're doing differential expression analysis. You might be given samples from different experimental conditions and look at the effect of treatment on gene expression levels, transcript abundance ratios, alternative splicing, transcript boundaries, and transcriptional start sites. So differential expression, there are a lot of tools out there that can do this type of analysis. So we can break down what they do into 5 basic steps. So first, we will align our reads, our RNA sequencing reads, to a reference. Next, we will count the reads that align to each gene or transcript or feature that we're interested in the reference. Then we'll normalize the read count. We'll discuss normalization in more detail later in the presentation, but this is going to be your FPKM and TPM values. Then we'll account for the variance in the read count. And after that, we're going to perform our differential expression analysis test. And that's where we're going to determine if the differences in read counts between samples or sample groups is significant. Next, let's talk about transcript assembly. What exactly is this? So if you consider the average human protein coding transcript, it has about 3 to 10 exons and is around 2,000 base pairs in length. If you consider the length of one of these protein coding transcripts compared to the length of library fragments generated during library preparation, the library prep fragments are an average of 150 base pairs long. So they're very short. And when we do RNA sequencing, we're only sequencing these fragments partially. So we end up with these relatively short reads in comparison with the original transcript length. What transcript assembly then does is it attempts to put these short reads from these short fragments back together into long transcripts. Now, novel transcript assembly is taking these short reads and assembling them into the long transcript without relying on any existing reference information, so it allows us to look for novel transcripts that may not be found in the existing annotation information. This is one of the analysis types that the Cufflinks app does really well. We won't go into a lot of detail, but we'll talk about it in a little more depth during the workflow discussion. So novel transcript assembly is a pretty complex process, but I'm going to break it down into 3 basic steps. So essentially, what's happening in the software is that we first take all of our reads and align them to a reference genome. Then after that, we're going to sort the reads by position that they've aligned to in the genome. After, we're going to take the sorted reads and create bundles of overlapping regions of aligned reads. And then, without relying on existing annotation information, we're going to try to assemble them into transcripts to try to identify novel transcripts in our data. So now we have an understanding of differential expression analysis and novel transcript assembly. Let's talk about some workflows under the RNA-Seq Alignment app that can perform these analysis types. Keep in mind, when using the RNA-Seq Alignment app to perform the full workflow, including differential expression, 2 apps are going to be required. The output from RNA-Seq Alignment gets used as input for the follow-up differential expression app. Also, I just want to put in a quick note that these particular workflows are going to be best suited for libraries that are prepared using TruSeq RNA-V2, TruSeq Total RNA, TruSeq Stranded mRNA, TruSeq RNA Access, and TruSight RNA Pan-Cancer. So here I've broken down each workflow by version and follow-up app. As you can see, different versions of the same app can lead to different workflows. Let's talk about each one in a little bit more detail. One thing that I do want to note is that it's not going to be possible to cross workflows. So what I mean by that, if you ran the RNA-Seq Alignment version 2.0.0 or 2.0.1 app, you're not going to be able to feed that output into, let's say, the Cufflinks NDE app. Okay, so let's talk a little bit about the highlighted workflow here. So our legacy TopHat Alignment app uses mainly the main components of the Tuxedo suite, which you may already be familiar with. So this workflow only supports 3 genomes. It uses only the TopHat 2 aligner, and it performs novel transcript assembly in the Cufflinks NDE app only, so only in the second app. This workflow, as mentioned, it's our legacy workflow. So if you're coming in new to RNA-Seq analysis, we would recommend using a newer version of the tool. However, this is intended for anybody that's continuing experiments that may be using these particular suite of tools. Next, we have these 2 workflows that are highlighted here. These workflows are actually pretty similar, so I'm going to mainly just point out the differences. The newer version, the RNA-Seq Alignment version 1.1.0 and 1.1.1, are really meant to support our targeted panels like the TruSight RNA Pan-Cancer, the TruSight RNA Fusion, and the immuno-oncology filter. Additionally, under the V1.1.1 app, this is going to support the newest data model under new mode in BaseSpace, which is going to allow for data aggregation. Other than those differences, these workflows are pretty similar. You're going to have your choice of different aligner between the TopHat Bowtie, TopHat Bowtie 2, or STAR. You can select one of 17 reference genomes, some of which are non-human. You can perform optional gene fusion calling with TopHat Fusion if you selected Bowtie or Bowtie 2, or Manta if you selected STAR. SNP and indel calling is going to be performed using the Isaac variant caller. Abundance estimations are going to be performed by Cuff Norm, And again, these estimations are going to be done in FPKM values. And differential expression will actually be performed by Cufflinks. Also note, these workflows do support novel transcript assembly, and we'll talk about that a little bit further, but the assembly will be broken up into the two apps. An alternative to the other workflow is to feed your RNA-Seq Alignment version 1.1.1 output into the DE-Seq2 app. Now, the DE-Seq2 app is a BaseSpace lab app. So what that means is that this app is one of our as-is apps, and support is a bit limited. However, of course, if you come with questions, we will do our best to address them. Now, again, in this workflow, because you are using the RNA-Seq Alignment app first, you're still going to be using one of the three aligners mentioned previously. You still have your choice of 17 reference genomes. The main difference here, though, is that differential expression is going to be done using the DE-Seq2 algorithm, which is version 1.1. So this is going to be gene-level expression only. Oh, and also our abundance estimations here are still going to be done using FPKM. Lastly, we have our newest workflow, which is highlighted here. I'm not going to go into a whole lot of detail since we're going to detail this further in the next portion of the talk, but I will point out that this workflow is significantly different than the previous workflows because we're going to be using TPM's transcripts per million for abundance estimation. So again, we'll talk about this further, but I did want to point out just a few other things with this. So this workflow, when it comes to the QC metrics, it's going to use the full data set to estimate these metrics. In the previous workflows, we were only using a subset, so the metrics for this app are going to look different and really shouldn't be compared with any metrics that you got with previous workflows. Also, please note that this new workflow does not actually perform transcript assembly. So before we detail the latest workflow, I just want to take a step back and discuss this workflow in slightly more detail. Mainly because this workflow does perform novel transcript assembly and differential expression. So if this is your goal, this is going to be the recommended workflow that you follow. Okay, so the RNA-Seq Alignment version 1.1 app is going to feed into the Cufflinks version 2.1 app, and this workflow together, again, is going to perform your differential expression and your novel transcript assembly. So I want to take a moment to talk about the different tools used by these two apps. As we've talked about already, these groups of apps are going to be fully using the Cufflinks suite of tools. So when we align our reads, again, we have our option of star, TopHat, and TopHat bowtie, TopHat bowtie 2, and TopHat bowtie and TopHat bowtie 2 are part of that Cufflinks suite. So if you make that selection, you will be including, you will be going down that path. Next, we're going to count reads using the CuffQuant app. The CuffQuant portion tool inside of the app. Then our normalization is going to be using CuffNorm and CuffDiff. We're going to account for variance with CuffDiff, and then we're going to determine differential expression significance with CuffDiff. Okay, so for novel transcript assembly, again, using this app, the main steps that we want to look at is that when we're actually going to divide the reads into bundles and assemble them into transcripts, that's all going to be done by Cufflinks. So Cufflinks is a piece of the software that gets used in both of the apps. So during RNA-Seq Alignment, we're going to create our novel transcript assembly for each replicate, and that's again using Cufflinks. And then in the second part of the app, the Cufflinks assembly and DE app, Tough merge will merge the individual assemblies for each replicate with the existing annotation to create a final assembly for each group, and to also point out our novel transcript. Okay. So now that we've given that option for you if differential expression and novel transcript assembly is your goal, let's switch gears and let's talk about our newest analysis. So let's detail our RNA-Seq Alignment version 2.0.1 app. So again, we're going to be following this specific workflow in just greater detail. So when you launch the RNA-Seq Alignment version 2.0.1 app on BaseSpace, you're going to see a form that looks just like this. This highlighted portion here, this will autofill by default with just the name and the date. However, if you want to rename this, you can do so right here. Okay. One moment. Okay. Then, in the following area, you can select a project for which you wish to send your output results to. In the next piece, you're going to select the biosamples or the FASTQ files that you're going to run through the app. Then we have our reference genome selection. So here you can select between the pre-built genomes, or you can select the custom option. If you do select the custom option, you're going to need to use the Custom Genome Builder first, and you're going to need to select the output of that app in the following fields highlighted here. Next, if you're using one of the panels for your RNA-Seq experiments, you can select that panel here. So the options are going to be TruSight RNA Cancer, RNA Fusion, or the Immuno-Oncology filter. If you didn't use a panel, you can just leave it at none. Next down the forum, you're going to see the alignment options. So if you select QC mode, that means that your metrics are going to be based on the number of read pairs that you select here. Otherwise, the metrics are going to be done fully against the full data set. If you check off the Call Fusions checkbox, this is going to be performed using Manta. If you have any of the ERCC spike-in controls, you're going to select that from the drop-down here. If you have genotypes of interest, or if you've uploaded a VCF with genotypes of interest, you can also select that here. Lastly, the advanced options, you do have the ability to modify these options. However, if you're new to this, we recommend running it with all of the default options. And then you're going to launch the app. So behind the scenes of the app, this is what's actually occurring. We are going to perform these different steps in the workflow. So first, let's talk about filtering and trimming the reads in the FASTQ file. From this, you're going to get your filtered FASTQs that would then get put into the alignment. So let's detail that a little bit more. So when we filter and trim our FASTQ files, we mean that we're going to align the reads to abundant sequences in order to get rid of them. So these abundant sequences are rRNA, mitochondrial RNA, poly-A, poly-C, SpiX, and any adapter. So this alignment is done with TopHat bow tie, and there is no option to change that. This is just the default for filtering your files. So the filtered reads are going to remove any abundant sequences from the FASTQs. And then also we're going to have a trimming step that trims the 2 base pairs from the 5' end of all the reads. So you're going to have your sample FASTQ files as input, and you can have your single read files or your paired end files. TopHat bow tie is going to be used, and we're going to filter and trim against the abundant sequences. And we're going to get our sample filtered FASTQ file. And then these FASTQ files are the ones that will be used in the alignment step. Okay. So let's move on to the alignment step, where we're going to actually get our BAM files for each sample. Okay, so we discussed this a little bit before. Alignment is performed with the star aligner. And you only have this option. So the star aligner is generally fast and performs well for the longer and paired end reads. Also, the star aligner does really well with any reads that you might have that have a lot of variance. So it will still be able to align correctly even if lots of variants are found inside of those reads. Star uses a pre-built star index within the specified reference genome, and it also performs self-clipping. So in our workflow, those filtered FASTQ files will get fed into STAR, and STAR is going to output, it's actually going to output 2 BAM files. So we're going to get our main BAM file and our main BAM index file, and this is a file that you can put into, let's say, IGB for further browsing. We're also going to get a transcriptome BAM file, and this BAM file is going to be used for quantification later on. We'll talk about that again a little more. And some other files that you're going to get from this alignment are going to be your coverage bed graph and your coverage in SIGWG format. So one more note here, because you're actually getting 2 BAM files, you're not just getting 1, your folder size or your actual output is going to be bigger than, let's say, if you ran a previous version of RNA-Seq, because you have 2 BAM files that account for a lot of the space that will get produced. Okay, so the next step is the optional fusion calling. And from here, if you do select it, what you're going to get is a list of fusion genes for each sample. So fusion calling is performed by Manta, and it does require that we have paired end reads. So our sample BAM files, the first BAM file, that's going to get fed into Manta. Manta is then going to give us our fusion candidates, and the fusion filter is what's going to give us our final fusion candidates in the CSV output file. So you actually get 2 outputs for this. You get the fusions.csv with all of your candidate fusions, and you also get a VCF file format for these fusions. The next step in our workflow is to call the variants, and from here we're going to get a VCF and genome.vcf file for each sample. So variant calling is done using the Strelka variant caller, which performs our small variant calling. It uses a specific random forest-based variant scoring model. And before doing the actual variant calling, we're going to remove any duplicate reads that would be found in the BAM file. Also, the annotation engine used for this is going to be Nirvana. So we're going to take our sample filtered BAM and then our reference genome, feed them into Strelka, and we're going to get 2 main output files. We're going to get our sample.genome.vcf file and our sample.vcf file. So just a few notes here about the differences between the genome VCF file and the regular VCF file. The sample VCF file is going to contain all of the variants that were identified, passing or not passing. So that means you're going to get your list and you're going to be able to see which variants actually pass. The genome VCF is going to contain all calls. That includes reference calls, any calls that were actually found to be the same as the reference. And again, you're going to get passing and non-passing for both the reference calls and the variants. Finally, our last step in this workflow is going to be the quantification and read counting. So from this, we're going to get our TPM values for all known genes and transcripts identified for each sample. Also, from the read counting step, we're going to get a list of raw read counts for each sample. So let's talk about quantification. We have a tool called Salmon, which quantifies expression of the transcript. This is where we're going to use that transcript SPAM that was generated during the alignment. We're going to quantify expression of annotated transcripts in each sample, and it's going to aggregate transcript level estimates to gene level estimates. Then it will assign the TPM values, or the normalized abundance estimate, for each gene and transcript. So this is going to be the main output that we're going to get from this app that is going to go in for our differential expression analysis app. So again, down here we have our sampled transcriptome SPAM, and we're going to use our filed transcript sequences, and we're going to feed that into Salmon, and that's going to give us our sample.quant.sf file and our sample.quant.genes.sf file. So because we're using TPM values instead of FPKM, I wanted to take a moment to just describe some of these differences. So you may be familiar with FPKM values, or fragments per kilobase of million mapped reads. In this version of the workflow, we're going to be calculating transcripts per million of mapped reads. Now, FPKMs and TPMs are similar, but the order of calculations is performed differently. When you're performing TPM normalization, we first normalize for gene length, then divide per million, then divide per million scaling factor. So when we use TPM, the sum of all TPMs in each sample will be the same. Because the sum is the same, it's easier to compare the proportion of reads that are mapped for each gene in each sample. In contrast, the FPKM, the sum of the normalized reads could vary, and it will be different. So if you want to stay with FPKM values, if that's what you prefer to use for your abundance estimation, then it's best for you to go back and use the RNA-Seq V1.1 workflow instead of this workflow. However, if you are interested in using this one, you're going to get these TPM values. Also, I've linked you up here to a really good tutorial that details RPKM, FPKM, and TPM, so you can clearly see the difference of how this different normalization works. Okay, so moving on to the read counting step. I bundled this with the quantification step, but really it is its own step. So in the read counting, all we're doing is we're counting the number of reads that align to genes in the existing reference annotations for each sample. So we're going to take our sample BAM and our existing gene annotation. We're going to feed it through the RNA-Seq read counter, and that's going to give us a sample.counts.genes file. So over here on the right, we see the names of the genes and the raw read counts that were assigned to that gene. So this output file doesn't actually get used by any of the apps. So all it is, it's mainly for us as a user so that we have an idea of the raw read counts that were assigned to each gene for each sample. Okay, so with that, our final output for the RNA-Seq Alignment app, which is going to be then used in differential expression, is going to be our quants.sf and our quants.genes.sf files. So I have here a preview of what they look like. Over on the left, we have the quants.sf, so that's going to have our transcript and our TPM values. And over on the right, I have our quants.genes.sf file, so that's going to have our genes, and it's going to have our TPM values for those. So lastly, I wanted to take a moment to discuss where you're going to find all the different output files for this sample. So for each sample. So when we are on our BaseSpace app session and we're looking at our final output, we want to go over to the Files tab. In the Files tab, we're going to see folders for each of the samples that you ran. In this example, I ran 4 different samples. And then when I click into the individual samples, I'm going to get a list of all the folders accounting for each of the steps in the workflow. So we have our alignments folder, counts, filtered. Filtered is really our filtered FASTQ files after removing abundant sequences. We're going to have a fusions folder if we decided to call fusions. We have our metrics, quantification, and reports and variants. So over on the left, I've just outlined what to expect in each of the folders, but we already discussed that in some detail. Lastly, I just wanted to show you the PDF summary report that does get generated. So this is a pretty comprehensive report, and it's going to use all of the main outputs, and it's going to basically put it in this PDF summary so that you have a quick way of looking at a sample, or of looking at statistics for each of your samples. Okay, so now that we've performed our RNA-Seq alignment, let's move on to our RNA-Seq differential expression analysis using our v.1.0.1 app. So like before, when you launch the RNA-Seq differential expression app, you're going to see a form that looks like this. Over here in this filter for RNA-Seq alignment section, this is where you're going to select the same preferences that you had picked for the RNA-Seq alignment app. This is going to allow the app to filter and look for results that match the preferences from the RNA-Seq alignment app. Next, you have the option to enable differential expression. So when you click this, the app will perform differential expression analysis between two groups of samples. If selected, both your control and comparison data sets must be specified. One note here about this, if you don't select it, the differential expression app will just perform global analysis. Next, if you do enable differential expression analysis, you also have the option of enabling paired sample analysis. So what this means is that we're going to perform analysis between two groups of samples, and if they have to be paired. So again, in the next part of the forum, we have our control group label, which you do have the option to change the name if you wish, and you do have your comparison label. So again, if you're not performing differential expression, if you're just doing global analysis, you're only going to have the option to select data sets under the control group. But if you are performing differential expression, then you are going to have the option to also use the comparison group label and select data sets for the comparison group. And if you used paired sample analysis, you are going to need to select data sets in a paired fashion. So here, in this example, what I did, I selected our control samples, which are going to be our UHR total RNA samples, and then the comparison group are going to be the brain total RNA samples. So that's the group that I'm going to compare against the UHR samples. And then we launched the app. So on the back end, what's occurring when we launch the app is that the RNA-Seq alignment output, those quantification results, are going to get fed into two pieces of the workflow. So when you launch it, global expression analysis is going to be performed no matter what for all samples. And then if you did select differential expression, that analysis will also be performed using DE-Seq version 1.2. Okay, so let's look at global expression analysis and what our output files are going to be. So here what we're mainly going to generate are the sample correlation matrix and the PCA plot. So let's talk about the sample correlation matrix. So here what we're going to show is the similarity of the samples based on correlation of the expression values. So the app is going to perform hierarchical clustering based on the correlations. The correlation levels are expressed in a range between +1 and -1, where +1 means that the samples are total positive correlation, 0 means no correlation, and -1 means a negative correlation. So here we have a small example of a sample correlation matrix. So as you saw in my example when I was launching the app, I did have my UHR samples and my brain samples in different selected. So what we see here So we first see hierarchical clustering. That's what this bracket here means, and it's showing you that it found that the UHR samples are similar, which we would expect, and that the brain samples are similar. And then together these two groups can also be considered as one similar third cluster. Now, when comparing this to our samples, we also have our heat map here showing our correlation. So for the UHR samples, when compared to themselves, we're going to see a total positive correlation, and that makes sense here. When comparing the UHR samples over with the brain samples, we do see a slightly less correlation, but we do see that they are somewhat similar. Next, the next output of the expression analysis is going to be our PCA plot. So our principal component analysis, or PCA, shows samples along the first 2 and 3 principal components that capture the most variance. So the app is going to give us both a 2D plot and a 3D plot if we have 3 principal components. So let's take a look at that. So over on the left here, I have our 2D PCA plot, which just shows our 2 principal components. And our principal component 1 here, this just means where the most variance, this accounts for where the most variance was found. And our principal component 2 accounts for the second most variance. So when we're looking at this plot, we see that 2 samples are close together here. Those are our UHR samples, and we can say that they're kind of forming a cluster with themselves here. And then over here, over on this side, we see our brain samples, and they seem to be right around this first principal component. And then they do have some variance when we're looking at the second principal component. Now, when we use the 3D PCA plot, we still see the same kind of pattern that we have here. It's just now that we're seeing it in this third dimension where we're considering the third most variance for each of these. Now, along with our plots, our heat maps, and our PCA plots, we're also going to get gene expression files. So first, we're going to get our normalized gene expression, or TPM. So that's over here on the left. And we're going to get our regularized log transformed gene count file as well. Okay, so if we selected differential expression analysis, we'll get the results of that as well. So one of the first results is going to be our heat map. So our differential expression heat map will show the level of expression of genes across various comparable samples. So similar to the sample correlation matrix, we are going to see some hierarchical clustering between the samples and also between the genes. And then the expressed genes are going to be assigned different colors. So anything in the red, this means that the genes have lower expression. Black means average expression, and then neon green means genes with higher expression. So here we have an example of our heat map, our differential expression heat map. Now, it's similar looking to the correlation matrix, except that now we have our samples across the top, and we have our genes across the side here. So we have the same hierarchical clustering that we noted before, where we have the UHR clustering together, the brain samples clustering together, and then that's creating our third cluster here. And similarly, we're going to have the same thing for our genes. So up at the top here, these 2 genes are creating one cluster, but since there are many, many genes, you're going to have a lot of different clusters, and they're going to be clustered based on how similar they are in this analysis. And then, of course, we have our color coding, so we can see here that this top gene for the UHR sample here is highly expressed, and there's low expression for this brain sample and average expression for these other two. Another output that we're going to get is the gene browser tool. So the gene browser shows an interactive scatter plot of the log2 counts of the genes for the control samples and comparison group samples. So basically what this is, it's going to plot all of our different genes, and we're going to get this type of image. Now, one of the best things that we can do with this particular gene browser, and I don't have a demo here, is we can select a significance and we can set it to true or false. So true being truly significant difference in expression, and false meaning not. And you would actually see which genes and how they separate across this log2 graph. And with that as well, you will get the same information in table format. So if you don't want to use the genome browser, you can still get the same exact information in this table format. And again, you can sort it by significance, and you can even download this as a CSV file so that you have your final results available. Okay, so let's switch gears and briefly talk about the RNA Custom Genome Builder. So one of the advantages, again, of this newer workflow is that you can do RNA-Seq expression analysis with a custom genome. So we're specifically looking now just at this app. So when we launch the Genome Builder app, we're going to get a form like we have seen before. These first three files, these first three fields are going to be free text fields for you to enter your species name, your provider, and your build. The whole genome FASTA field, you're going to have to select your .fa file for your custom genome. And this must have been previously uploaded. So that means that you will have to upload the .fa file into a project so that you can select it for this app. Next, you can select your abundant sequences. So again, during that abundant sequence step, that filtering step, you can provide your own FASTA files for the abundant sequences that you wish to remove. Additionally, if you have other abundant sequences that you want to remove and you're using, say, an HG19 genome, something that's already pre-built, you would still want to go through these steps to ensure that you have those selected. And of course, you need your GTF file as well. And again, this will also need to be previously uploaded. And there are advanced options that you can select for how we're going to index this with the SAR aligner. But again, if you're kind of new to this, we suggest just keeping everything the default. So when the app completes, you're going to go into the files and you're going to see a sample with the naming convention that you selected. You can click into it and you're going to see an annotation and sequencing folder. And if you click into the sequencing folder, you'll see whole genome FASTA and you'll see your actual genome file. So then when you select this in the actual RNA-Seq Alignment app, you're going to select custom for your reference. You're going to select your data sets under your custom options. You're going to select the genome that you built. And then it's going to appear like this in the RNA-Seq Alignment form. Okay, so with that, let's go ahead and wrap up and start discussing some other RNA-Seq analysis apps and tools that we have available. So again, we've thoroughly discussed the RNA-Seq Alignment workflows, and again, these workflows are going to be geared for libraries that will be prepared with the TruSeq RNA, TruSeq total RNA, TruSeq stranded mRNA, basically these whole transcriptome type of analysis. But you may have different types of RNA-Seq libraries that you'll want to use different apps for. So here in this table, we've identified our other options, and we've just outlined what we have available. So one app to point out here is the RNA Amplicon app. So if you have any type of targeted RNA panel, the RNA Amplicon app is what you're going to want to use. So this app is going to have, it's going to perform alignment using DWA. It supports Amplicon panels only. It does perform gene fusion calling. You do have your option of 12 different genomes, and it does perform differential expression using DESeq2. So this is going to do gene-level gene expression only. Also, we do have our Dragon RNA pipeline. So With this pipeline, you're going to use the RNA-Seq alignment using Dragon RNA-Seq Splice Aligner, so it's a very specific aligner. You have your option of using 5 human reference genomes. There is also the option to build your own custom genome, but you're going to have to use the Dragon Reference Builder, very similar to the custom reference builder that we showed before. It is going to perform gene fusion calling if you select it. And it's also going to perform quantification, and again, this quantification is going to be in TPM, not FPKM. Okay. So lastly, I've linked you up to some additional resources listed here. So we have our online help guides for both the RNA-Seq Alignment and the RNA-Seq Differential Expression app. I do have a resource here on the STAR Aligner in case you're interested in learning more about that. We do have our MANTA resource so that you understand how fusion calling is performed in more detail. Again, I've linked you up to the TPM discussion from StarQuest. And we also linked you up here to some additional tutorials on PCA plots and heat maps using hierarchical clustering here. So with that, let me move on to some questions.